today I'm actually going to talk about the two halves of my laboratory from the perspective of the precision medicine initiatives that many other diseases have been very heavily invested in, but specifically looking at how we can use some of these initiatives to help guide some of the mechanisms that we discover in cardiovascular disease. So precision medicine and cardiovascular disease, essentially I'm going to be looking and answering two specific questions. The first is, just so we're all on the same page, precision medicine, what is it, why is it believed to be useful, and what is its role in cardiovascular disease currently? And the second is, how can initiatives that are looking at precision medicine and omics-based research be used to inform mechanisms of disease and perhaps the role that we play in what we understand in cardiovascular disease? So what's the definition of precision medicine? What is it, and how is it believed to be useful? Well, the US NIH defines precision medicine as an emerging approach for disease treatment and prevention that takes into account individual variability in genes, environment, and lifestyle for each person. While some advances have been made, the practice is not currently in use for most diseases. So essentially, the idea, if you're looking at the cartoon, is currently a lot of medicine is one size fits all. But if we can stratify it, we can figure out which groups of patients are better served by certain types of treatments and interventions. Precision medicine essentially takes that one step further so that we can look at an individual and know what's best for him or for her. So I like to think of this sort of on a simple scale. If you're looking at one-size-fits-all medicine, it's the advice, don't put salt on your food, okay? But not everybody doesn't have to put salt on their food. So the stratified medicine is measuring a biomarker, which is essentially blood pressure, and deciding who needs blood pressure control with a therapeutic. In precision medicine, we would be able to figure out, you as an individual, what's the right therapeutic specifically for you, and should you be eating salt or not be eating salt? So we can really, rather than giving general recommendations to our patients, we can understand the specific patterns and mechanisms to help guide individual therapies. So what's the difference then between precision medicine, personalized medicine, and pharmacogenomics? So there's a lot of overlap between the terms, and they're often used interchangeably. But really, personalized medicine is considered an older term. And it's sort of not being used as much because the idea is when people hear personalized medicine, there's a misinterpretation that something is being applied or is discovered uniquely based on them. It's not that personalized medicine couldn't exist, but when we try to make things be more precise, we tend to study larger populations and hone in. We don't tend to pick one person out of this room, study them extensively from head to toe to give them a personalized assessment. So although the term is falling out of favor, it's possible in the future we will have more personalized medicine. Some of the send-away genetics, genomics companies essentially are providing a form of personalized medicine to people, obviously with a lot of flaws and issues, but in theory you could still have personalized medicine. Pharmacogenomics is the study of how genes affect a response to a particular drug, and it's really part of personalized and precision medicine. So it's a specific area of it. It often gets used interchangeably because a lot of the discoveries have been in pharmacogenomics. So what are the limitations? Well, the limitations are really manifold. The first is we often don't have enough information about a situation or disease that we care about. Many technologies are at early stages of development, so there's technological challenges. And then there's problems, I know from my own work, in having large amounts of data. Okay, and how do you study that data? How do you manipulate that data? There's ethical, social, and legal issues. How do we protect participants' privacy and confidentiality? Obviously, it's been very difficult to do that, and it's hard to reassure a lot of people who participate in studies to try to be proponents of precision medicine because data often gets leaked. There's also obviously a cost. There are issues of drugs that are currently the way they've worked in terms of the pharmaceutical industry is they can be applied broadly to people. What's the incentive to a pharmaceutical company to narrow down the focus of how their therapies are being used? And then really healthcare providers and researchers really need to know more about both molecular genetics and the biochemistry that's being overlaid onto people's information. So this is a summary table from Dan Longo that he had done in the New England Journal about 4 or 5 years ago that goes through a list of different medical fields. You could see here the diseases, the biomarker used in the intervention. What's sort of interesting about tables such as this is whereas with cancer, a huge amount of progress has been made. With hematology/thrombosis or cardiovascular disease, this is, even though this is a little bit of a dated article, this is still about as good as we get, really sort of highlighting how we haven't quite kept up for various reasons with other fields. So the current state of precision medicine is really, if you're looking at it globally, it's really been defined as genetics and its use in oncology. That's been the primary successful use of precision medicine. 73% of the biomarker trials have been in oncology. So that's the field where there's been the most traction and the most success, and success sort of breeds use in a field. 94% of pre-selected stratification type trials have also been in oncology. So there's really been a focus. This slide is just really saying that, you know, although certainly the European Medicines Agency and to a lesser degree the US FDA really want people to be banking DNA specimens when they're doing clinical trials, they're not providing necessarily the support to give more information. And as I just spoke about, in oncology, there's a different paradigm in terms of the encouragement of the pharmaceutical industry into developing therapies that can be tailored in this way, as opposed to cardiovascular, where it's very multifactorial and very broad, and there's less motivation. So for cardiac disease, there are many limitations of precision medicine. CVD is not cancer. Cancer is obviously a lot of different things. CVD often, especially for vessel wall type diseases, tends to get lumped together. We've obviously not nearly had the success with GWAS. You know, there's a lot more going on in terms of specific areas in the deep sequencing and using, you know, multifactorial type, which is very much precision medicine, to try to add to the risk factors to predict people who are going to have cardiovascular disease. But clearly, it's not had the predictive risk and intervening ability that other fields have had. So what this sort of suggests is that on top of what we find for genetic risk, we need to superimpose data that might add additional information on top of what we know clinically, on top of the standard imaging or tests. And this can include many other things, additional family history, the microbiome, epigenetics, and we need, especially for cardiovascular disease, in as many patients as possible. So for cardiovascular disease, we've asked, how do we get better precision? And this is just a slide sort of highlighting the fact that for cardiovascular disease, currently we have our markers. We measure blood pressure. We take a family history. But the idea is, can we come up with something that would be the equivalent of a liquid biopsy? And that could include, when I say liquid, it may not be full-out liquid. So it could include microbiome, swabs. It could be something from the saliva or something from the plasma. Is there accessible tissue or specimens that we could use to better define cardiovascular disease? In our own work, in my research lab, so my research lab is essentially two halves. We have a basic half that uses animal models and cell culture models to study atherothrombotic disease, and the other half uses high throughput, different types of techniques. We do a lot of transcriptomics, a lot of low abundant transcriptomics, and we do proteomics, and we partner typically with the Broad at MIT to do metabolomics. So the first piece of information, I'm not going to focus on metabolomics in this talk, but I'm going to give you some hot off the press data from metabolomics because I think it highlights ways we could use these data in sort of interesting ways. So one of the problem is when you measure, and I'll show you some of these data, when you measure things in people who already have disease, it's hard to get a lot more traction in predicting who's going to have an event on top of standard cardiovascular risk factors. the talk is about using those information to understand mechanism. So I think I'm still in the clear. You don't all have to get up and leave because I'm not going to tell you something that's important. But what's interesting about this metabolomics project was my collaborator Ravi Shah, as part of a grant we work on together, decided to look at the metabolites. Now, there's been metabolomic screening in populations like Framingham and older individuals when they already sort of are in the age range where you'd get cardiovascular disease. But he asked a question, can we look at some of these things that have been looked at in, 55, 65, 75-year-olds? If we look earlier, will that predict cardiovascular risk? So what he did was he took about 2,400 young adults, so mean age of 32, and did LCMS profiling, so big metabolomic profiling screen. And he came up with unique signatures of metabolic health. And this was a good population because it's divergent by race and sex and ethnicity. And he looked at these different profiles. He has 20 years follow-up, and we were able to validate it in the Framingham Heart Study in a slightly older cohort. So he looked at this metabolomic profiling. He came up with risk scores based on what was expressed. And he looked at this, and he came up with what he called a metabolite health score. The paper isn't completely written yet, so I'm not sure we'll leave it at that, but that's what we're calling it right now. And what was very interesting is even if you control for the common cardiovascular risk factors, if you look at a group this young, before people go on to develop hypertension or diabetes, because my RNA-focused brain and not metabolomics-focused brain always thought, well, metabolomics, it just means you're overweight, or it means, you have diabetes, and it's not really telling you anything, or renal disease, it's not really telling you anything new that you don't know. But in fact, people who don't have any of that, people who are not overweight yet, you can tell from their metabolomic profile, and that's what this is the vascular health score, and this was his myocardial health score, so who will have heart attacks. You can cluster these people and predict, independent of that, when they're in their early 30s, who in 20 years has a much higher risk of having disease, suggesting that we maybe can bump back the risk that we're using our surrogates, actionable things, high blood pressure, weight, diabetes, we can bump that back to even early to intervene on people. So that's what's so exciting about these data. They may or may not work as an overlay of what we already have, but they may work in different ways, and that's what's very exciting. So what else could we add for greater quote-unquote precision? We've been very interested in looking at transcriptomics. And one of the reasons we got very interested in transcriptomics is we are very interested in low abundant RNA sources because a lot of the work we've done is in platelets, so we've had to optimize these sorts of technological methodologies. And what's very interesting is when you look in really, I don't know how to put this, any liquid that comes from your body, so we look at urine, we've looked at CSF, we've looked at breast milk, we've looked at plasma, so plasma is the big one. And if you freeze those things and thaw them out, there's a very stable pool of RNAs in them. It's not high abundant, it's low abundant. What it is actually doing from a mechanistic perspective, I don't have time in my talk to put in, but we've been able to show passage of these RNAs and that they can be functional, especially if they're interacting with the platelet. But how does these stable pools of RNA relate to cardiovascular disease in different populations? So it's known that vascular cells release microparticles, and a lot of these particles are coming from platelets, but it's very difficult to tell when you're actually analyzing these particles where they're coming from because they tend to shed certain surface markers that you pick up by flow, and then they merge with other cells, and then a lot of times they're going through the platelets trafficking, and the platelets leave a few of the receptors. So where they're actually coming from is very highly contentious. So all we could do is say they're vascular ones that have probably been shared by several cells. The other thing is there's also non-vesicular-based RNA, and these are bound to lipids or proteins. And so these are very, very common low-abundant sources of RNA as well. So this is just a summary from a summary article that we did last year trying to split apart, the big particles from the little particles, and we tried to split it apart further from the lipid and the protein bound. One thing I will say as a result of having written this is there is very little consensus in this field. So those of you who are getting into this field and not already in it, just be warned that no matter what you say, there's going to be someone who's going to disagree about whether what you're claiming is in a cell is in there. All I will say is I think they're highly dynamic, and they are more dynamic in states of inflammation or disease, but they're found in many, many sources. It's not even clear why they're always there. We think they're a form of intracellular communication, kind of a slow boat way. They're not super fast, but a slow way. They've already been shown probably to be important in metastases. We also think, especially in regards to the platelet, they're a form of waste. Because we've been able to track them and see that the platelets love to suck up some of these things and then take them away where they get cleared in the spleen or the liver. So they're a source of not housekeeping genes, but have actual virtual body housekeeping. So why is it interesting to add this layer of extracellular RNAs in? Well, we sort of felt it would provide a bridge between sort of the genome and the protein. It really sandwiches itself. I'll present in a little bit. We do look at cells too, but one of the reasons we're very interested in these non-cellular sources is they're very stable, and you can find large populations to study. And the question is, if you're looking at something that's precision medicine type, or you're asking that sort of question, you're not asking the question to find one gene. You're asking, you want a multi-panel of genes. Just like with the metabolomics, it's a cluster of genes that's going to help define disease. And then the next question is, if you're asking a question about a disease, where should you get your source from? So if you're studying something, is it okay to get it from a certain source or tissue? Now, for everyone in this room who does any kind of basic science, that was a sort of a stupid question because, of course, everything is different. But I deal a lot with population scientists. And to them, if you've studied a panel, let's say you've sequenced whole blood, they think that's interchangeable with sequencing their white cells or sequencing their plasma or sequencing their urine. They use it's from a person. And it's a very fundamental concept, but it's one that sort of needs to be brought up with people. So I think the highlight of why this is true was the GTEx study, which was done across the river over at the Broad also, where they did RNA sequencing from 960 cadavers, fresh cadavers, and they did 52 different non-diseased human tissues, essentially, from each of these cadavers. It was quite a tour de force. And so it's a public repository of data, so if you're studying kidneys and you want to know what gene expression is like typically in this big group of people in kidneys, you can look. But one of the things that they did was they looked to see how the gene expression clustered. And clearly, obviously, as anyone in this room would expect, gene expression in a kidney is going to be different than gene expression in a brain. Again, this is now whole tissue. No one is isolating out cells. But it just highlights how different sources are going to be. So there's not too much coding RNA in a liquid biopsy. So this I know because I know these people very well. This is all coding RNA. If you ask them about microRNAs or lincRNAs, they can't help you. This was all done sequencing for coding RNAs, and also shows you one of the problems with technology. We can't seem to keep up with it and provide the correct data for people, but this was done prior to people caring about microRNAs. They didn't redo the sequencing twice to do small RNAs in addition to long RNAs. So when you look at these liquid biopsies, there's not much coding RNA. So if we sequence plasma, you do by sequencing get hits of coding RNAs, but if you spend any time looking at what the sequences are, you learn, especially because we often validate everything with PCR, you learn that almost all of the coding RNA is pieces. So most of the long RNA in these sources is pieces of long RNAs. So if it's matching to, pretty much anything, it's a piece of it. It's not a whole thing. And there's been a lot of, I think, myth perpetuated in the literature that people find these whole RNAs. It doesn't mean that from a precision medicine perspective that finding, IL-15, a piece of it doesn't reflect some sort of disease process or predicted disease process, but what it does suggest is there's probably not mechanistically usable IL-15 floating around in your microsome. So it depends, in your microparticles. So it really depends why you're getting this information. That's why that's important. So because of that, and because we've been very disappointed in the coding RNAs that we've seen, we've gotten very interested in these sources in non-coding RNAs. There's a lot. We've started out very heavily. A lot of the data I'm going to show is microRNAs, because that in a lot of these sources is what shows up. But there's many, many different types that you see when you start doing sequencing, and I'll touch on that also at the end. And they're there for various reasons. Some of them are easier to figure out why they might be important in disease, like microRNAs. And some we find piRNAs, tRNAs, all sorts of SNO RNAs, harder for us to understand if it's meaningful. And it always takes work back in the basic lab to figure out if you're just wasting your time looking at some of these things. So one of the populations that we've studied the most heavily and published on the most is from the Framingham Heart Study. And the reason for that is actually a physical proximity. I originally, back in 2002 or something like that, wanted to study platelets. And I somehow convinced the Framingham Heart Study, when they enrolled one of their cohorts, that they would let me set up a lab, which was about 30 minutes outside of Boston. And now it's even closer in Worcester. And we would isolate platelets, white cells, plasma, red cells. They had never done that before. And that this was very early in the transcriptomics thing. It was before sequencing. But they allowed me to have a lab out there for almost 3 years while they enrolled a whole cohort. So we enrolled about 2,500 people. We got isolated cells done by my person in the lab, who does a great job, because a lot of it is technological. If you mess up your sample preparation, especially for RNA work, it's not as robust as proteomics and metabolomics are. So it was the 8th visit of the offspring group. So the offspring group are the children of the people where they discovered that high lipid levels, high cholesterol is associated with cardiovascular disease back in the 50s. So their kids now are at their 8th visit at Framingham, and they're about 65. And we enrolled all of them and the Omni cohort, which is a minority cohort that matches in about age. And over the years, in this 8th visit, and now we've done it in a couple other visits and in their children, we've done a huge amount of gene expression profiling. I'll touch on the platelets in a second, but we've done whole blood, plasma, we have urine. I can't begin to present it all, because if anyone has questions, please feel free to, I always say we have way more data than we know what to do with. I get funded to generate the data, and then I really need other people to use the data, because you can only ask so many questions on it. So, and then since then, we've worked with dozens of other population projects. So as a basic scientist, I never thought that I would be doing this. It's a very strange thing to be doing. But I'll explain to you why the parts I like the best out of it. So in this particular project, we have sequencing in the laboratory, and it was a study where we took 40 people from Framingham, 20 who had cardiovascular disease, 20 who didn't. We took their plasma, we sequenced it. We came up with a list of targets, and we validated about 800 targets by high-throughput PCR it's 2,700 people who are in the regular cohort and another 400 in the minority cohort. So about 3,100 people. We did high-throughput PCR, which is much more reproducible than sequencing and super fast. Like the isolations take a while, but we churn through that amount of PCR, millions and millions of reactions in under 2 weeks. So we have a platform set up with robots doing the pipetting, and everything is good. It was a U award at the NIH, so we're on a really short time scale. Okay, so I have to make decisions, and this will be in the last slide. You'll get why, because you're going to think I'm really stupid or crazy. But I had to decide from the sequencing which 800 things we were going to target in under 2 weeks, which was pretty daunting, because you've got these giant, and we made a lot of mistakes. But one thing we did do is we discovered it's still probably to this day the largest sort of mass screen of short RNAs in the literature. And most of it, as you would expect, were microRNAs. We found some SNO-RNAs. We found some piRNAs that people debate, are they really piRNAs? I'm not going to get into that. And we found that they mostly associated with age. Different patterns as you get older, a little bit with gender. The race ethnicity thing is very complicated. We've done a bunch more populations, and there are some differences in race and ethnicity, although they're less than you would think. So what did we do with this big data set? Well, one of the things we're very interested in, as you might guess from the previous metabolomics slide, is we're very interested in obesity, insulin resistance, and diabetes. And the question is, some people have high BMIs and are fine, some don't. So the BMI sort of centric definition of obesity doesn't necessarily predict what's the underlying biology. So what we did is we looked at this big group of people, and we have some validation groups to go along with it. And interestingly, we found that 18 specific RNAs, small RNAs, were associated with insulin or HOMO-IR, which is another way of looking at it for people who aren't in the field, but also fraught with some contention. And we looked at the association with adipokines. We have CT images on a lot of these people looking at their visceral and hepatic fat, so we could look at subcutaneous visceral fat, hepatic fat, so we could see if it associates with different fat depots. We found that there were specific microRNAs that were very highly associated with visceral fat, with the development of diabetes. And then interestingly, we were able to go back in a group of almost 100 children from a population at Children's Hospital over at Harvard, and found that in these children, even if they weren't fat, if they had the expression pattern similar to the people who developed disease, it was starting to show up in children as young as 2 years old. And in that population, we were able to do both some proteomic and metabolomic screening. And this is just a background slide from that Jane Leopold and Joe Lascalzo did in Circ Research a couple of years ago. We did what was called an interactome. So basically, we all have an interactome where our genes are associating with our proteins, our metabolites, blah, blah, blah. And then if you look at it and blow this up, there's an area, a little circle in that interactome of our body, that's billions of interactions, that is related to a disease, and it's called the disease module. And what's interesting about this sort of philosophy of looking at diseases and how it might be interesting to use later in precision medicine is that you can take common different phenotypes of people, cluster them out to different networks, and figure out who's low risk and high risk. And we were able to do that with the information from these various populations looking at their metabolomics and their microRNAs primarily, and some of their genes. And we really find when we did this overlay of nodes that it turns out that this one particular microRNA is really in the center of all of it. So some of these things, when we do these interactomes, which are done by our collaborator at UCSF, Alex Pico, sometimes it's very similarly weighted for certain diseases. For this, it was actually very centrally organized around miR-122. And some of our collaborators are now looking at potentially developing therapeutics related to miR-122 activity. Just going to inject a little of my own bias. I'm not a huge believer in terms of disease prediction of single microRNAs. In my own experience in the basic side of the lab, when you change things, you don't ever typically change one microRNA, you change a whole cluster. And there's nothing wrong with studying the mechanism of that one mir, but I think you have to be reflective about that it's not like a single protein coding gene that might be lethal. It's going to be a family and cluster of mirs typically that changes. And sort of related to that is using these data to look at heart failure. And the idea to look at heart failure was that, current algorithms that we use to predict heart failure don't really capture who's going to develop it. And there's really a consensus now that most heart failure, especially if it's not single genetic sorts of heart failure, is very dependent on a multi-hit. And that's many of the models of preserved EF-type heart failures are following this even in their animal models. So we looked at that specific population and we found 12 specific extracellular RNAs that seem to travel together and predict the development of heart failure. And to do this group of people, we also had LV mass and other echo parameters. So these people have also, this is where you need to understand these sort of complex relationships. You need a really densely characterized phenotype population. And one of the problems with Framingham is they're all white, but they've had everything done. So if you can get a hold of those data, it's one of the richest data sets that we get. And their operating procedures are perfect. Like they save their blood perfectly. They do everything they're kind of crazy over there. But it's helpful to your science. So what we found is if you're looking at these echo parameters that relate to heart failure, these 12 extracellular RNAs were associated. When we first measured this, we didn't have follow-up on these people, but time has gone by. And we actually have the development of 116 cases of heart failure that developed during 7.7 years of follow-up. So now we can look at this and see who develops heart failure. And we did so adjusting for age, sex, and people who had prevalent MI. And what fell out after that were 3 microRNAs. And these microRNAs, I'll show you the figure, were associated with a 20% reduction in long-term incident heart failure per 2-fold increase. And what's very interesting about these 3 microRNAs is there's a lot of homology, there's a lot of sequence homology. So as you would guess, they predict many of the same things like TGF-beta and many things that you would already guess are important for the development of potentially of heart failure. And this is just the one for 106B5P. And so if you have a higher level of this particular microRNA, you'll have decreased LV hypertrophy and remodeling when looking at all 2,300 people it's 2,300 because that was how many we had echoes on. And a decreased risk of developing heart failure over 8 years. And for bioinformatics, as I said, hypertension was the main thing that lit up. We're collaborating with people who do a lot of the heart failure mouse models over at the Mass General, and this is just sort of a sampling of some of the culture and animal models that they've done. They've looked at the effect of gain and loss of function of these 3 microRNAs, and so far it's very consistent with what you would expect that we're seeing in the humans. And the idea is to figure out if it's specific to one of the microRNAs and what happens in combination. Do we see more in combination as well? So what about other sources of RNA in disease? So I keep telling you about platelets, so that's what I'm going to talk about. As I said, we've done urine and fecal matter and saliva, but my basic lab likes to, even to this day, study platelets, so I'm going to just focus on that. And platelets were originally the reason we got involved in the Framingham Heart Study. I had the ridiculous idea of wanting to measure RNA and platelets in 3,000 people, and as I said, they agreed to it. And the original study that we published from these data back in Circulation in 2010, essentially what we found was that platelet gene expression is very different in patients who have cardiovascular disease, especially if it's related to obesity and diabetes. And the genes that went up, and they're highlighted across the bottom, are interestingly not genes related to things that you would think cause thrombosis, like the platelet surface receptors or things that would make your platelets sticky, but are related to immunity and inflammation. So higher levels of these things typically, although for a couple of them they're lower. And what was interesting is what also lit up were all the pathways. So it was a really easy first story because it was receptors and all the signaling pathways downstream all lit up kind of together in select individuals. And it was very dependent on obesity and diabetes. But what we also found interesting was that so I'm going to skip over a lot of the things that we did with this, but we have found that all of the innate immune receptors are found quite widely in platelets, which seemed like a kind of crazy idea when we originally found this back in around 2005. Why would there be innate immune receptors in platelets? They are not innate immune cells. And this is just a listing. The other interesting thing is it was observed in my lab that we years ago saw very big differences between male and female mice This is the great thing about having both halves of the lab. We were able to immediately go over and recapitulate what we found in mice and humans. So it was not just an artifact of the mice, it turns out, and it's kind of very germane now with everything going on with, you know, the coronavirus and the differences between men and women. We saw very different patterns of innate immunity in women and in men. And so that was, and it was all actually derived from careful observation in the mice. So this is just a summary figure of sort of the family of innate immune receptors that people are aware of. And the first one that we actually studied was toll-like receptor 2, because it's actually a bacterial one, but it's very heavily expressed in platelets. And I'm not going to show you all the stuff because we published all sorts of stuff about this years ago. But what's very interesting and highlighted from pictures rather than figures is, so here's a resting white cell and some platelets. And in this test tube are isolated platelets, and they make a cloudy liquid because they're like snowflakes that float around. And if you add thrombin, which would be like if you cut yourself and you're clotting off, it turns into this kind of crazy spaghetti stuff because it's sealing the wound, right? Or it's causing a heart attack, you know, whatever. It's causing a clot. And all the stuff floats to the bottom and it gets cleared. If you stimulate through toll-like receptor 2 or a bacterial pathogen, you don't get either of these things. You get the platelets hanging out outside the white cell, but they don't look like they're forming a thrombus. And if you look at this, it looks more like resting. So it's very interesting. This was actually an observation a graduate student made in my lab. He was like, it doesn't seem to be working. It looks right, but it doesn't look right, and I don't understand it. It turned out, when you stimulate this way, the platelets really stick to white cells. So by flow cytometry, everything changes, right? It doesn't change by an aggregometer, which measures platelet sticking, but you see all of this platelet white cell sticking, which now seems, for people in the field, seems like, well, of course. But back in 2005, we had no idea what was going on, right? People hadn't even really described the receptors that cause platelets to stick to white cells very well. So we just saw this, we saw the pictures. It's very interesting. So after looking at things in bacteria, we've actually started to look at some of the other receptors. Now, I initially ignored these because they're in the endosome, and I thought, well, because I'm really simple, I said, you have an infection. Here it is. It's on the surface. It's not in the endosome. Why do I care about things that are inside the platelet? I don't care. But we started to get a lot of hints that it was very, very important, and we started to look at some of these endosomally located ones. And just so you know, this is a picture of a platelet that has it's probably a CMV-based family of viruses. You can see that on this big old white cell, the platelet is stuck to it. And we like to say, if you turn it, it looks like a beret, actually. But you could see the little viral particles. And you can like visually see the interaction of the platelet with the white cell and what it's doing by EM. So what we decided to do is look at the interaction of these endosomally located ones, which are virus receptors. So we started looking at virus receptors. This is just a summary of some data from a paper about 5 years ago where we looked at toll-like receptor 7. And essentially what Milka Kupanova showed in this was when you inject animals with relevant viruses for toll-like receptor 7, what ends up happening is you get a whole lot of heterotypic aggregates as reflected by this figure and this decrease in platelet counts, and you can see the time course. But if you have a mouse model that doesn't have toll-like receptor 7, you don't see it. So it's very toll-like receptor 7 dependent for viruses. What's also interesting is with the bacteria, we do get some clot formation, but with these particular viruses, we weren't getting clot formation. It was entirely platelet white cell interactions. And the animals that don't have it die much more commonly and faster from viral infection. So it's important for fighting off the infection as well. So a lot of this has become even more relevant because there have been actually, this is an article from about a year ago in the New England Journal of Medicine. There was actually one 3 or 4 years ago that's fairly similar, not quite as honed in on influenza, but that there is a risk of influenza, of myocardial infarction for the first week after you have a flu infection. And this increases for about 7 days, and then it tends to normalize out. And it's one of the main reasons people die of flu. And Milka, for she ramped it up last year because it kind of depends on how bad the flu is each year. She had been collecting blood from acute flu patients at our hospital, which is right next to where our research building is. And so she had been studying all of these flu patients specifically. And what was very interesting is, flu's not widely thought to be freely circulating around, but she clearly started to see when she was doing some imaging, the flu is green and the platelets are red, that there was flu very close to what it looks like, almost on top of the platelets or trafficking with the platelets. And based on this observation, we did do some EMs, and in a certain number of patients, not all of them, we were finding particles within these locations. These are probably, it depends exactly what stage you're looking at. Some of them are endosomal locations or lysosomal locations in the platelets. It looks like there were viral particles inside some of these platelets from real people who were having flu infections. So what she did was she took flu and she incubated platelets with flu and showed over time course that, in fact, in a purified system, you absolutely see platelets taking up flu infection. So my bias that I don't want to look at a receptor that's inside the platelet was obviously completely wrong, because the platelet's taking it up, putting it in, right into that, triggering one of those toll-like receptors to have a downstream effect, and this is just the time course. And this is showing some very nice high-resolution images of the flu particles going inside of the platelets. What was very interesting is when she now took this information that she got from the patients, and that we now had definitions of what was happening to people in populations who have TLR7 or don't have TLR7. She looked at this and found that what looked like it was happening is if the platelet got a hold of the virus, the viral particle, it was doing something to speak to the white cell, which was then causing the white cell to have DNA release. Okay, so DNA release from the white cell can lead to thrombosis formation downstream. So it didn't look like it was probably a direct interaction of the virus with the platelet, because the platelets weren't shooting out their pseudopodia to look like they were activating. They were communicating with the white cell, which was causing histone and DNA release. And this was, I'm not going to go into all the data, but essentially her hypothesis of what was happening was flu was getting inside the platelets, triggering TLR7, through direct interactions and complement C3 release, activated the neutrophils. The neutrophil activation led to DNA release and net formation, and we were actually able to see what almost looked like nets in some of the patients with flu, and that in some people is going to lead to thrombosis. So when I think about using omics and how it's kind of changed how we've looked at things in the basic lab, the original hypothesis and the original working paradigm we had for platelets was this, that a platelet gets activated, it releases coagulation proteins, chemokines, cytokines, and it causes hemostasis, it stops you from bleeding, or thrombosis, it causes you to have a heart attack. But through these large omics projects and looking at disease outcomes in people, we actually have changed a lot of what we do in the basic lab, because now we realize that platelets are very, very important for immunity and inflammatory reactions. So the last little bit I'm going to show, and I'm only going to show this slide because I have all sorts of crazy things. When we looked at that original set of 40 people and then expanded it in another population and looked at the sequencing data, showing the strengths and the negative parts of sequencing data, we found a lot of crazy stuff. We had a very advanced pipeline because we were part of this NIH program, which threw it up, after it went through the human part, it threw it up against all sorts of other species. And we were seeing all sorts of kind of crazy stuff. And some of the crazy stuff is just crazy. Like, why would people have algae or tobacco or soy? We got very excited when we saw the food things, because in Framingham we have food diets. So the first thing I did was, if you have high expression of soy microRNA, are you a soy eater? And the p-value on that was off the charts. But when you looked at the data, it turned out it was being driven by 2 people who ate more than 30 servings of soy a week. So that's not, so 2 people made our p-value. I got really excited, and then you have to drill down. But these things are kind of, these things are both intriguing mostly probably wrong, but kind of crazy. And one of the things we have started to look at in these sort of other categories is it turns out some of the pollen ones are real. We've been able to, validate it in mouse models where we've taken pine pollen and given it, in inhalation chambers, been able to get the microRNA from pollen to go up. Part of the problem with that whole project, it was a lot of work. It's a lot of work to convince people you have pine pollen mirrors. The other thing is it doesn't do that much to the animals, which makes sense, right? Because otherwise you go for a walk in the trees, you go for a walk in the spring, and you would have something different happen to you, right? Other than happiness and, uplifting moments. So that's why that whole project was really fascinating, but is sort of a tough sell because it doesn't show anything particularly earth-shattering. But since in a lot of these people, we also have long RNA sequencing, we're looking at lincRNAs. The lincRNA thing, we are actually doing, that entire group, we're doing lincRNAs in it. Why lincRNAs are tough if you're looking not mechanistically and you're looking at a population is a lot of them are pieces of the lincRNAs. So you may get a big screen for lincRNAs, but you're not seeing the whole thing. So what does that really mean? And so it's the discrepancy between sequencing and the next step, which might be PCR. Other people are very interested in circular RNAs. We're looking at those in these populations. I don't know what layering all of these things will mean. So in summary, when considering precision for cardiovascular disease, more broader, reproducible, and cost-effective information is obviously needed. We want to predict disease, but we may need to look earlier at what I call actionable stages. So, there's earliest molecular detection, initiating events, but we may need to be looking back at baseline risk to have actionable areas. But it is possible we may be able to better predict with our current therapies who is best suited. So in summary, what are the potential benefits? Well, hopefully we'll have improved ability to predict our treatments and our patients and ourselves, and better understanding of the underlying mechanisms, which obviously can help in many, many ways in terms of developing new treatments and diagnostics. And then we also, as kind of an off-target, is we need new approaches to protect research participants and privacy and confidentiality. We need, really need tools for analyzing and sharing large sets. This is probably the biggest. I never would have thought this. I would have thought generating the data was the hard part. The hardest part is figuring out the right way to use the data, the right way to share the data, that kind of thing. It really requires new partnerships. Our work in this has led to my collaborating very far outside my field. I work with, I think the most extreme thing I work with is hibernating bears. So, yeah, so poor Milka had to go up to Michigan to track bears when they were hibernating and get their blood. So you could, yeah, it's everywhere, it's everywhere. But it is the opportunity for, millions of people to contribute to the advancement of science and be engaged in new treatments and therapies. So I first want to thank everybody for being here. I want to thank thank you for this wonderful invitation, and the great people who've helped me with this, particularly Carmen Tanner-Verity, who runs the High Throughput Lab. He's amazing. Milka Kupanova, who did a lot of the later platelet innate immune work, and Ravi Shah, who is central in all of these population study projects that I work on. So thank you very much.